Hey everyone, this is Steven and welcome to my channel, and today we're going to talk about transcription from DNA to RNA. But before that, don't forget to like and subscribe my YouTube channel. So let's start. So in genetics, transcription is the process by which the polymerization of ribonucleotides, which were guided by complementary base pairing, will produce an RNA transcript of a gene. The template for this RNA transcript is one strand of that portion of your double helix that composes the gene. So this is technically transcription comes after the DNA replication, which is performed by different enzymes within the mechanism of your cells. RNA polymerase is an enzyme which is synthesizing a single-stranded RNA copy of a gene. And in the next figure, you will see the basic components of transcription, and it illustrates key events in the process as it occurs in the bacterium Escherichia coli. And the figure will be divided into different transcription phases, which is initiation, elongation, and termination. That will be calling the end of initiation. Here, this is your this electron micrograph, and this is the promoter, and simultaneously all of these RNA polymerase, okay, or the transcription bubble are elongating, okay, starting to transcribe simultaneously. And geneticists often use the direction of travel by the RNA polymerase as a reference when discussing various features within a gene. If, for example, if we started on the 5' end of a gene at point A and moving along the gene in the same direction as RNA polymerase to point B, you will be traveling in the downstream direction. And if by contrast, okay, the opposite of that direction is what we call as your upstream direction. Termination is another process in transcription where a hairpin loop is being formed, okay, as a termination signal, and the RNA polymerase will be released at a terminator. So let's talk about this in the further slides. This information flow again is collinear from DNA going to after the transcription and going to mRNA strands. So this is your primary transcript to be continued to process in your translation. Okay, so let's talk about termination. Termination is the end of transcription. RNA sequences that signal the end of transcription are what we call as terminators. There are two types of terminators. We have intrinsic terminators, and it will cause the RNA polymerase core enzyme to terminate transcription on its own. So it's more likely to have a committed RNA polymerase to stop the transcription. The other one is extrinsic terminators, which however require proteins, okay, other than RNA polymerase, and they will require a polypeptide known as the Rho, which will bring out termination. All terminators, whether that will be intrinsic or extrinsic, are specific sequences in your mRNA that are transcribed from specific DNA regions. Then terminators often form this what we call as the hairpin loops, in which the nucleotides within the mRNA pair with nearby nucleotides. Upon termination, okay, RNA polymerase and a completed RNA chain are both released from the DNA. And the product of transcription, again, is a single-stranded primary transcript. The RNA produced by the action of RNA polymerase on a gene is a single strand. We call them as a primary transcript. And the bases of the primary transcript are complementary to the bases between the initiation and the termination sites in the template strand of the gene. Okay, and the ribonucleotides in the primary transcript would include the initiation codon or the start codons. These codons are specifying the remaining amino acid of the polypeptide and the stop codon. So that's how we are gonna define the single-stranded primary transcript, which is a product of transcription. The enzyme RNA polymerase again catalyzes transcription. DNA sequences near the beginning of the genes is what we call as promoters. They will signal RNA polymerase to begin transcription. So as you see in the bacterial gene, promoters share two short regions which have almost identical nucleotide sequences, and these are the sites at which RNA polymerase makes particularly strong contact with the promoters. RNA polymerase adds nucleotides to the growing RNA polymer into the 5' to 3' direction, and the chemical mechanism of this nucleotide adding reaction is very similar to the formation of your phosphodiester bonds between nucleotides during the RNA replication, but with one exception. And transcription are using these different ribonucleotide triphosphates, ATP, CTP, GTP, and UTP, instead of deoxyribonucleotide triphosphates. And hydrolysis, if you can still remember, of your high energy bonds in each ribonucleotide triphosphate provides the energy needed for elongation. And fourth, sequences in the RNA products known as terminators will tell your RNA polymerase where to stop the transcription. So you'll see here a low temperature electron micrograph of a cluster Escherichia coli bacteria magnified 10,000 times, and each individual bacterium is oblong shape. So this transcription initiation signals in bacteria. In this figure, you will be seeing promoters of 10 different bacterial genes. Only the sequence of the RNA strand is being shown, and numbering starts at the first transcribed nucleotide 11. Okay? Most promoters are upstream of the start point of transcription. This one is upstream, this one is downstream, and all promoters and Escherichia coli share two different short stretches of the nucleotides, which are shown in yellow, that are essential for recognition of these polymerases. Okay, they are the most common nucleotides at each position will each stretch and constitute a consensus sequence. They are in bold, so this let strong Escherichia coli promoters and their consensus. So these are 10 different bacterial genes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay? And these are the bold letters are what we call as your consensus sequence. They are the same among all your bacterial genes. So in summary of the first part of transcription, although the transcription of all genes in all organisms roughly follows a general scheme shown in the figures 8.11, it's very important to note this variation. For example, the transcription of different genes in bacteria can be initiated by alternative sigma S factors. In eukaryotes, the promoters are more complicated than those in bacteria, and there are 3 different kinds of RNA polymerase, and they can transcribe different classes of genes. RNA polymerase is what we call as the key enzyme of transcription, and they recognize the promoter at the beginning of a gene and then uses this complementary base pairing with your DNA template, okay, to add the RNA nucleotides to the 3' end of your growing transcript. When the RNA polymerase will detect a terminator, it dissociates from both the DNA and the transcript. And thus, this is what we call as your extrinsic termination. If it detects a terminator, if it stops on its own, it's what we call as the intrinsic termination. So let's talk about the other domain of life, the eukaryotes, okay, and how does transcription process in the multicellular organisms, or majority of the multicellulars. RNA processing after transcription will produce a mature mRNA. So since RNA processing in eukaryotes modifies only the 5' or 3' ends of the primary transcript, it will leave the information content of the rest of the mRNA untouched. Other processing deletes blocks of information from the middle of the primary transcript, so the content of the mature mRNA is related but not identical to the complete set of the DNA nucleotide which is paired in the original gene. Okay? Mature mRNAs are related but not identical. So we will be adding okay, the process in eukaryotes is adding a 5' methylated cap and a 3' poly A tail. So the nucleotide at the 5' end of a eukaryotic mRNA is a G in reverse orientation from the rest of the molecule, and it is connected through a triphosphate linkage to the first nucleotide in the primary transcript. So this backward G is not transcribed from the DNA. Instead, there is a specialized capping enzyme that adds it to the primary transcript after the polymerization of the first few nucleotides. So this is being added, this capping enzyme is being added. These enzymes, known as the methyl transferases, then add a methyl CH3 groups to the backward G and to one or more of the succeeding nucleotides in the mRNA, forming a so-called methylated caps. Okay, this is what we call as your methylated caps. It has a methyl group. This is your guanine, okay, and these are not transcribed. This triphosphate bridge then linked to this transcribed bases. So this is a structure of the methylated cap at the 5' end of a eukaryotic mRNA. So this capping enzyme connects to the backward G to the first nucleotide of the primary transcript through a triphosphate bridge. Methyl transferases enzymes will add methyl groups to this G and to one or two of the nucleotides first transcribed from your DNA template. So in this figure, okay, figure 8.14, like the 5' methylated cap, the 3' end of most of the eukaryotic mRNAs is not encoded directly by the gene. In a large majority of eukaryotic mRNAs, on the other end, the 3' end consisting of 100 or 200 A's, and we referred to as a poly A tail. Addition of the tail is another two-step process. First, a ribonuclease will cleave the primary transcript to form the new 3' end. Cleavage depends on the sequence of AAU-AAA, which is found in poly A containing mRNAs, which are 11 to 30 nucleotides upstream of the position where the tail is added. Next, the enzyme poly A polymerase will add A's onto the 3' end and exposed by the cleavage. So this is how an RNA processing adds the tail to the 3' end of your eukaryotic mRNAs. So this is a ribonuclease will recognize, okay, AAU, AAA in a particular context of the primary transcript, and then cleave the transcript 11 to 30 nucleotides downstream, okay, downstream to create a new 3' end. So this, once the ribonuclease will recognize this AAU, AAA, then it will add a poly A polymerase, okay, add A's into the 3' end of this strand, and there we go. We got a poly A tail on the 3' end. It's around 100 to 200 A's. So let's talk about removing introns, okay, from the primary transcript by what we call as the RNA splicing. Another kind of RNA processing became apparent in the late 1970s after researchers had already developed that enabled them to analyze nucleotide sequence in both DNA and RNA. Using these techniques, they began to compare eukaryotic genes within the mRNAs derived from them. And a very extreme example of this length difference between primary transcript and mRNA is seen in the human gene for dystrophin. In this figure 8.15, we will know that abnormalities in the dystrophin gene will underlie the genetic disorder, the Duchenne muscular dystrophy, or the DMD. The dystrophin gene, for your information, is a 2.5 million nucleotides, or that's 2,500 kilobases long, whereas the corresponding mRNA is roughly a little 40,000 nucleotides or 14 kilobases in length. In the late 1970s, the researchers had developed techniques that enabled them to analyze the nucleotide sequence in both DNA and RNA of this dystrophin gene, because they've noticed why is that the gene, okay, has 2,500 kilobases, wherein the mRNA is reduced in, okay, has added like 14,000 nucleotides, or just 14 kilobase in length. It has reduced its number. Okay, let's talk about muscular dystrophy, the type. This photo on your left is the histopathology of gastrocnemius muscle from patient who died of a pseudo-hypertrophic muscular dystrophy, Duchenne type. We will see a cross-section here that the muscle shows extensive replacement of muscle fibers by your adipose cells. The picture on your left by Dr. Edwin Ewing Jr. is a drawing of a 7-year-old boy with a Duchenne muscular dystrophy. There is an excessive development of the lower limbs. We call that one is pseudo-hypertrophy, and thinness of the arms. In this figure, you will see that there is a lumbar hyperlordosis is very visible from a drawing of a 7-year-old boy. So let's talk about dystrophin. Dystrophin is a rod-shaped cytoplasmic protein and is a very vital part of a protein complex that connects a cytoskeleton of a muscle fiber to the surrounding extracellular matrix through the cell membrane. And this DMD gene, which encodes the dystrophin protein on your right, is one of the longest human genes known. It covers 2.3 megabases at locus of X in your XP21. So, the primary transcript in muscle measures about 2,100 kilobase and will take 16 hours to transcribe. Okay, the mature mRNA measures 14 kilobases, and the 7-9 exon muscle transcripts codes for a protein of 3,685 amino acid residues. And now, we'll take a look at this human dystrophin gene, which is an extreme example of RNA splicing. This dystrophin gene is around 2,500 kilobase long, and the dystrophin mRNA is only 14 kilobase long. More than 80 introns are being removed, okay, from the 2,500 kilobase primary transcript to produce the mature mRNA. So this is your dystrophin gene. This is your introns in yellow and exons in brown. In your primary transcript, it has 2,500,000 nucleotides, and after RNA splicing, the mRNA has more or less 14,000 nucleotides, and that's a big chunk that is being removed. Okay, the introns have been removed from the primary transcript going to your mRNA. So let's talk about exons and introns. What are they and how are they useful in the transcription process? Sequences found in both a gene's DNA and the mature messenger RNA are called exons. We call them as expressed regions. The sequences found in the DNA of the gene but not in the mature mRNA are what we call as the introns, or the intervening regions. Introns interrupt or separate the exon sequences that actually end up in mature mRNA. In the case of the DMD gene, it has more than 80 introns, and the mean intron length is 35 kilobase, but one intron is amazing 400 kilobase long. So these introns or intervening regions should be removed. And there is a question: how do cells make mature mRNA from a gene whose coding sequences are being interrupted by introns? The answer is that cells first again make a primary transcript which contains all the gene's introns and exons. Okay? It's a mixture of exons and introns, and they remove the introns from the primary transcript by what we call as the process of RNA splicing. They will delete the introns and join together successive exons to form a mature mRNA, which is consisting of only the exons. And this mechanism of RNA splicing will be shown using these three types of short sequences within the primary transcript: splice donors, splice acceptors, and branch sites, and they will help ensure the specificity of splicing. These sites, okay, in this mechanism of RNA splicing make it possible to sever the connections between an intron and the exons that precede and follow it, and they will be forming and joining all the separated exons. So these are the short sequences that dictate where the splicing will occur. We have the splice donor, branch sites, and splice acceptors. So the splice donor site occurs at the 3' end, okay? Splice donor site, 3' end, this one, of the exon, and abuts the 5' end of the intron. This is the exon, this is the intron. Okay? And most splice donor sites, a GU, okay, or a dinucleotide, this one, this two nucleotides that begin the intron is flanked on either side by a new purines. It's either purines that is A or G. Okay? Second, the splice acceptor site is at the 3' end, okay, this one, of the intron, where it joins the next exon, and the final nucleotides of the introns are always AG. So this is, if this is your start of your splice donor GU, it's the start at the end of a splice acceptor, it's always been AG. Okay, and it will be preceded by 12 to 14 pyrimidines. It's either C or U, and the third one is a branch site which is located within the intron, and it is having nucleotides upstream of the splice acceptor, and it must include an A, okay, this one, the branch site, which is usually rich in pyrimidines. So this is your splice donor, branch site, and splice acceptor. And then there are two sequential cuts. The first is in the splice donor site here, and the second is in the splice acceptor site. And then they will be forming a lariat. After forming a lariat, then intron is being degraded here and joining the exons as a mature mRNA. So next we'll be talking about snRNPs or SNRPs and the spliceosomes. Splicing normally requires a complicated intranuclear machine. We call that one as your spliceosomes, and they ensure that all of the splicing reactions will take place in concert. And the spliceosomes have 4 subunits. We call that one as small nuclear ribonucleoproteins, or the snRNPs, or the SNRPs. Okay? Each SNRP is containing 1 or 2 small nuclear RNAs, or the snRNAs, which is about 100 to 300 nucleotides long, and they are associated with proteins in a discrete particle. Ribozymes, on the other hand, is a remarkable enzyme, okay, because of the complexities of a spliceosome structure. It is very remarkable that a few primary transcripts can splice themselves without the aid of a spliceosome or any additional factor. These rare RNA transcripts function as what we call as your ribozymes, the RNA molecules that can act as enzymes and catalyze a specific biochemical action. So this is photo on your structure on your right is a 3D structure of your hammerhead ribozyme. So splicing again is being catalyzed by what we call as spliceosomes. On your left, spliceosomes are assembled by 4 snRNP subunits. Each of them will contain 2 snRNPs and several proteins. So 5 snRNAs plus 50 proteins will form 4 snRNPs, small ribonucleic particles, which assemble into your spliceosomes. And then photo on your right is a view of 3 spliceosomes which are captured using an electron microscope. So let's talk about alternative splicing. What is alternative splicing? it's a different mRNAs from the same primary transcript. Normally, RNA splicing again joins together the splice donor on this portion and your splice acceptor at the opposite ends of the intron, and results in the removal of that intron and then fusion of the two successive and now adjacent exons. But for some genes, however, RNA splicing during development is very regulated so that at certain times or in certain tissues, some splicing signals may be ignored. For example, splicing occur between the splice donor site of one intron and splice acceptor site of a different intron downstream. So that may be sometimes ignored. Such alternative splicing produces different mRNA molecules that may encode related proteins with different, though sometimes partially overlapping, amino acids and sequences and functions. In effect, then, alternative splicing can tailor the nucleotide sequence of a primary transcript to produce more than one kind of your polypeptide. And alternative splicing is explaining us how 20,000 to 30,000 genes in the human genome can encode hundreds of thousands of different proteins estimated to exist in human cells. So in mammals, this alternative splicing of the gene encoding the antibody heavy chain determines whether the antibody proteins become embedded in the membrane of your B lymphocyte that makes them or are instead secreted in the blood. So this antibody heavy chain gene has 8 exons and 7 introns. Exon number 6 has a splice donor within it. To make the number bound antibody, all exons except for the right hand part of the number 6 are joined to create an mRNA encoding a hydrophobic C terminus. So let's check how it is being done. So this alternative splicing, of the primary transcript for the antibody heavy chain will produce mRNAs that encodes different kinds of antibody proteins. This orange is an outside of antibody heavy chain gene. This brown is for exons. This yellow is for introns. This blue one is an intron in membrane bound or exon in secreted. A is your poly A addition sites, and these red lines are splice specific for membrane bound. So after transcription, the primary transcript will splice for a membrane bound antibodies. All of your exons are found here, including the A tail. These exons 7 and 8 will encode a membrane attachment domain. So after this after they being transcribed, this will produce a membrane bound antibodies. This other one is splicing for secreted antibodies that flows into the bloodstream. So 1, 2, 3, 4, 5, 6A, and including the 6B, which is surprisingly an intron, but it's included in the primary transcript, and a poly A tail. And this will be a part of a secreted antibodies which flows in, again, your bloodstream. So different mRNAs from the same primary transcript, is all about your alternative splicing. And a very rare and unusual strategy of this alternative splicing is seen in Synapcarditis elegans and a few other eukaryotes. We call this as trans-splicing, in which the spliceosome joins an exon of one gene with an exon of another gene. So we'll take a look at that in the next slide. And these special nucleotide sequences in the RNAs makes trans-splicing plus possible. Now we'll take a look at trans-splicing and how it combines exons from different genes. So we have chromosome A and chromosome B. This is the leader exon, and this one is chromosome B with 1 and 2. So this chromosome A and chromosome B and all of their exons, leader and 1 and 2, are being joined together in okay, one mature mRNA, and they are coming from different genes. Okay, so that's what we call as trans splicing. It's trans, which means coming from a different source, getting an mRNA strand out of chromosome A and B. So in summary, we've learned that in eukaryotic cells, RNA processing follows a transcription to generate mRNA. And these processing steps include additions of a methylated cap to the RNAs at 5' end and a poly A tail to the 3' end, as well as removal of introns from the primary transcript by what we call as your splicing. Alternative splicing of exons can yield different mRNAs from the same primary transcript. Okay, that's all we have for today for transcription, and in our future videos, we will be talking about translation. I hope you've learned something from this lesson, and thank you so much for listening. Goodbye.